So today we are going to talk about meiosis. Now, you guys have learned about meiosis, but in little detail. What you guys have learned that in meiosis, the parent cell, that is a diploid cell. Then that parent cell divides to form two cells, which are both have the half number of chromosomes, and then each cell further divides to form total of the 4 haploid daughter cells. Now, these 4 are daughter cells and they are all haploid. Now, this stage where the parent cell divides into 2 cells and the number of chromosomes become half, this is referred as meiosis 1. And second stage where these 2 cells they divide to form total of the 4 daughter cells which have the haploid number of chromosomes. This stage is called as meiosis 2. Now, this meiosis 1 is a bit complicated, only up till the prophase 1 stage. Only up till the prophase 1 stage. And meiosis 2 is exactly like mitosis. It is exactly like mitosis. There are few things which you are going to learn today which are new. Now, if you guys remember, when you guys learned about mitosis, we learned that it is a cell cycle. Once a cell divides by mitosis, 2 cells are formed. Those 2 cells again grow and again divide, right? But this is not a cycle. This is not a cycle. Once these daughter cells are formed, they have no capacity, no capability to further divide. Question is, why? Now, in mitosis, from 1 cell when 2 cells were formed, 2 daughter cells were formed, let me show you that this was 2N cell, the parent cell. It divided to form 2 daughter cells. They were also diploid. So they can further go into the mitosis. These cells are haploid, already half the number of chromosomes. What half they can do? Not anymore, right? So it's impossible to go further, you see, beyond this stage. Another thing, these 4 daughter cells, which are made at the end of the meiosis 2, they specialize to form gametes. They specialize to form gametes. Now, in males, they change into sperms. And in females, they develop into ovum. Now, if you pay attention to the words which I spoke and which I wrote, I said that these 4 develop into sperms. It's plural, right? And in meiosis, in women it takes place, only 1 ovum is formed. Only 1 ovum is formed. Then question is, it means that out of these 4 cells, only 1 cell will develop into ovum. What will happen to the rest of these 3 cells? Now, that topic of oogenesis was in the old syllabus. It's not in your syllabus anymore. Otherwise, you would have understood that while this meiosis takes place, in meiosis 1, from this cell, when 2 cells are formed, this is 1 cell, 1 smaller cell is formed and 1 larger cell is formed. This cell also has single M nucleus. Now, this smaller cell is called a polar body. Now, this cell survives and this cell dies. The question is, rather this cell survive and this cell die. Question is, why does this cell die? Because this cell does not have all the cellular organelles. Well, this cell has all the cellular organelles. Then this cell further go where? Into meiosis 2. Then it will form what? A normal cell and other polar body, and this one will also die. So only one cell survives, and this cell develops into what? An ovum. Understand? So these smaller cells, what are they called? Polar bodies. This is just for your knowledge. This is not in your syllabus now. It was in the old syllabus.
Sir,
Yes.
it's a disease or it's natural?
No, it's natural. You have learned in 1 month, in a menstrual cycle, only one egg cell is produced from one ovary. If this is the October, and if the right ovary produces an egg cell, next month left ovary will produce the egg cell. Only one egg cell is produced at one time, right? How it happens? This is the procedure. When, sorry? Yeah, yes. That is an exception. Some, you see, some problem, something, right? So remember that this is meiosis 1. 2 cells are formed, right? But one of the cells, it became a polar body and it died. Why it became a polar body? Because it did not have all the cellular organelles. A cell to survive, it must have all the cellular organelles in it. Yes.
Why do the relationship between that and that?
Obviously, because you see, it is a hormonal effect as well, and they have to, you see, in 1 month, if the right ovary is performing this job, so next month it will take. Because remember one thing, you see, it's not in your syllabus, otherwise I have taught you. You see, in Pakistan, you go onto the walls of the street, is written, Hakeem Lukman, Mard kabhi booda nahi hota. Have you seen this advertisement in Pakistan? It's true. If we think about it, it is true. Why? Because since puberty till death, spermatogenesis never stops in men. They keep on producing sperms. It means that they can father babies.
Doesn't stop when you're 60.
Sorry?
Doesn't stop when you're like 50, 60.
No. In men, even 100 years old man is able to father a child. But a woman, they have a specific number of follicle cells. After the age of 40, for some women, you see, it is between 40 years to 50 years of age of the women when the follicle cells they finish. It's different for different women. The number is different. Once the follicle cells are finished, that stage is called menopause. No more formation of the egg cells. She cannot mother a child. Right? But the man at 110 years old age still gets married and can have children. Right? So it is true, when it's written on the wall, you never become old. Right? So this is one thing, right? That in women, you see, only one ovum is produced, but in men what will happen? All 4 will develop into sperms. If you guys remember your lecture of O levels, reproduction chapter, in one ejaculation, there are 250 million sperms. Right? How it becomes possible? Because every meiosis, every cell is developing it into a sperm. Every cell is developing into a sperm. Now, so main purpose of the meiosis is the gametogenesis. We call this process as gametogenesis, formation of gametes. Right? In women, it will be called as oogenesis, and in men, it is called as spermatogenesis. Formation of sperms is called spermatogenesis. Formation of eggs or ovum is called oogenesis. Understood, everyone? Why there is requirement of meiosis? Who will answer this question? You have prepared. Very good. To retain the normal diploid number of chromosomes. Now, if let's suppose there is no meiosis, if the sperm cells are made by mitosis. There will be 46 chromosomes in the sperm. And egg also is formed by mitosis, 46 chromosomes. When they will join 92 chromosomes, it will not be a human being. Sorry, right? So to retain the insanity, to retain the normal number of chromosomes, N and N, they will join together to make what? 2N. Normal diploid number of chromosomes. Understood? Next. The beginning stages of the meiosis are just like mitosis. Means that there will be a G1 phase. Now, who remembers from last year's lecture what was G0 or G1 phase? Very good. Where simple growth was taking place of the cell. Cell was increasing in size and the organelles, they were also increasing in size and number. Organelles. So in G1 phase, what is there? Increase in size of cell, 1 point, and increase in number of organelles. After G1 phase, there is S phase. What happens during the S phase? DNA replication. So over here, replication takes place. Duplication of the DNA takes place. Now remember one thing, when it is S phase at that time, DNA is not in the form of chromosomes. It is in the form of what? Chromatin. Loosely packed. It is loosely packed. Understand? After S phase, again there is what? G2 phase. Again growth takes place, right? And then starts the meiosis. Then there is M1 phase. We call it as meiosis 1. We represent it like this. Right? And then there is M2 phase. We represent it like this, meiosis 2. Understand? And then specialization takes place to form the either egg cell or the sperm cell. We are going to start with the meiosis 1. Now, who remembers that in mitosis there were 4 phases? What was the first phase? Prophase. Second one, metaphase. Third one, anaphase. Fourth one, telophase. All these 4 phases are over here, right? But in meiosis 1, we call them as prophase 1, metaphase 1, anaphase 1, and telophase 1. Now, in the beginning, I told you guys that prophase 1 is bit complicated. It is a bit complicated, but many of the stages, they match like mitosis. We are going to talk about that, right? As prophase 1, you see, remember that all the processes that are taking place in our body, they are continuous. They are continuous. Only for our understanding, for making it easy for us to understand and learn, we divide them into stages, right? Even prophase 1 is divided into 5 stages, but we are not going into that detail. That's not in your syllabus, right? So we are leaving that. We are keeping it simple. First of all, we are going to learn about prophase 1. Now, let's suppose that this is a cell. Now, this is a cell right after the S phase. This is the cell in G2 phase, right? So this is what? This is its nucleus, and its nuclear membrane is intact, if you remember, right? I will be showing you guys two chromosomes. I will be showing you guys pairs of two chromosomes, and we will be discussing some other things as well over here. So let's suppose that this is the chromatin right now. And over here, what is this? This is centrosome. This is centrosome. Now, before I move further, another concept I need to give you. That concept you guys have learned before as well, but I need to revise that for you guys. Homologous pair of chromosomes. Yesterday I did talk about it a little bit, right? I told you guys the word homo means what? Same. Logos means what? Information. Next thing is what? Pair. Now let's suppose I told you that yesterday that this is a chromosome. The second chromosome in the pair will be exactly the same. Now which thing will be exactly the same? The shape. First of all, the shape of the chromosome will be exactly the same. Now let me tell you, the shape, the position. Now you know that in the middle there is what? Centromere. Centromere is always not in the middle. Centromere is always not in the middle. There are 4 types of chromosomes actually. There are 4 types of chromosomes on the basis of position of centromere. On the basis of position of centromere. We are not going into that detail again, right? But this position can be different. In a homologous pair, the position of centromere will be exactly the same. Second, that they will have exactly the same gene on the same locus. Yesterday I told you what is a locus. Who remembers what is a locus? Very good. Location or position of a gene on a chromosome is called its locus. Now, let's suppose if over here is the gene for height, exactly at the same place there will be gene for height. Exactly the same position. Understand? So, they will have same genes at same loci. Understand? Now, this is one pair of chromosome before S phase. This is a pair of chromosomes before S phase, right? Now, when the S phase will take place, after that what will happen? Each chromosome will have 2 chromatids, right? Now, this is called one chromatid. This whole chromosome has one rod, right? This is one chromatid. This is second chromosome. It also has what? One chromatid. We have discussed this last year, if you guys remember. Now, when the replication has taken place, these both chromosomes can be represented like this. One and the other one. Right? So this one, these are what now? These are 2 chromatids. These are also what? 2 chromatids. Yes.
Can you explain this part again?
Yes. This is before S phase. Means that this is the chromosome during G1 phase. In the S phase, what happens? Replication. Each chromosome will make its copy. So when this chromosome will make its copy, it will become like this. So 2 copies are now attached with each other. In the center, where centromere is there, where centromere is there. Same, this is one chromosome in the G1 phase. When the replication will take place in the S phase, it will become like this. It will become two chromatids, two chromosomes joined with the middle. The chromosome number has become what? Double in the S phase. Understood now? Another thing. Now you see, this is a complicated diagram. I'm going to draw a simpler diagram now. Please pay attention. Let me draw over here. I can show this chromosome like this. Simpler way, right? And the other one like this. Now remember that these both are what? Homologous. Which are these? Homologous. Now, these two chromatids which are bind with each other, these two chromatids will be called as sister chromatids. The two chromatids of a chromosome, when they are bind together, they are called what? Sister chromatids. Same, these two will also be what? Sister chromatids. Understand? But this one and this one, they are not bind together, right? But they are from the homologous pair. They will be called as what? Non-sister chromatids. So after the S phase, when the chromosomes, they are in duplicate stage, two chromatids which are bind together, they are called what? Sister chromatids. Easy or difficult? Easy thing. But the chromatids from the other chromosomes, not from the same, right? They are called as what? Non-sister chromatids. What can be the reason behind it? This is your question. What can be the reason behind it? The reason is, you see, these both will be an exact same copy of each other, right? This one and this one is both exact same copies, replica. And these two are exact copy of each other. Now, I told you that let's suppose that there is a gene of height over here, right? Now, let's suppose that this one had a dominant allele. So these both will have what? Let me show over here. Capital H over here. And let's suppose the red one had what? Recessive allele. So these two will have what? Small h over here. Understand? So there can be difference. I said can be, not exactly, right? But for this one particular gene I'm talking about, for this one, there are thousands of genes on each chromosome. At some point, maybe one of the allele is dominant and other one is recessive. So they are not exactly the same, right? Because of dominance and recessiveness. So that's why these two will be called what? Non-sister chromatids, and these two will be called what? Sister chromatids. Yes.
Are they still homologous?
They are homologous. See, the gene is of same trait. Gene is of the same character, right? Height. But dominant and recessive doesn't matter. Got it? The height is one trait. Yes.
So it can be different genes. It has to be the same type of gene.
Yes. Different. See, I will explain you more inshallah about the allele as well. You see, yesterday we discussed the definition of allele. What was that? Counterpart of a gene is called an allele, right? Another definition for this is a different version of a gene. Different version of a gene. Height is one version. Sorry, the tallness is one version and shortness is another version. But the trait is exactly the same, height. Understand? Skin color is one trait. But there are many phenotypes of it, right? And those are because of different genetic makeup, different genotypes, right? Dominance or recessiveness. Understood everyone? So now you have got the concept of what? Of homologous chromosomes, sister chromatids and non-sister chromatids. Understood?
So that one in G1 phase also called homologous chromatids.
Exactly. This is also homologous pair, this is also homologous pair. But here what has happened? Duplication. Replication has taken place. Got it? Now, this is the during what? Let's suppose this is during G2 phase, I said. Nuclear membrane is intact. Here we do not have chromosomes, we have what? Chromatin. Remember, I told you, I have taught you this last year as well. Replication can only take place when the chromosomes are in which condition? In chromatin condition. In open form. Right? Otherwise, in the chromosome form, they cannot be replicated. This over here, this is what? Centrosome. Right? I am not drawing other organelles because we don't require them to understand during the cell division. Right? Now, what happens first of all? In the mitosis, what did you learn? Nuclear envelope starts to disappear. Remember that or not? Centrosome makes its copies, right? And each copy starts traveling towards the opposite pole. Remember that or not? Same exact thing is happening. Same exact thing is happening. Another thing, that this chromatin starts to condense and takes the shape of what? Chromosomes, right? So let me write down these points which are exactly same like mitosis. Centrosome makes its copy and centrosomes start moving towards opposite poles, right? Nuclear envelope starts to disappear, right? Chromatin condenses and takes the shape of chromosomes. Yes.
So these are the chromatin form.
Yes, these are no. These are chromosomes form.
But this is-
Chromatin. This is chromatin.
But this is before this phase.
No, I said that you see before S phase, but if I compact them, they will become like this. They will become like this. You see, the replication happened during the S phase. They are loose, but if I compact them, they will become this shape. Right? Now, you will understand now. What I am going to do, I am going to show that the same cell now in the early prophase. What do we call it as? Early prophase. Now, you guys can see what has happened. Centrosome has made its copies. Centrosome is moving over here in this direction, and this one is moving over here in this direction, going to the opposite poles. Next thing, I am going to show that one chromosome is this one, another one is this one. This is one homologous pair, right? The other one I will make the smaller one. One this one, and other one this one. So this is one homologous pair, this is another homologous pair. Understand? Next step, what I am going to show you. I told you, another thing, what happens that nuclear envelope starts to disappear. How will I show you guys the nuclear envelope disappearance. I told you yesterday.
What did you say?
Yesteryear. It's exact same word. You can check the English dictionary.
That's a word?
Yes. Like yesterday, yesteryear.
It's per week.
Now, so nuclear envelope is what? Dissipate. What happens next? That these centromeres, they reach to the opposite poles and bivalent is formed. New fancy word. Bivalent is formed. Tetrad is formed. Another word. Let me tell you, it's not difficult. Very easy. Now, nuclear envelope but disappearing. I am showing like this, right? The centromeres, they have reached where? To the opposite poles. What happens? That these chromosomes, which are homologous, they come close to each other. And they arrange as pairs. They arrange as pairs. The homologous chromosomes, they come close to each other and they arrange as pairs. Now, how many chromatids are here? 4. So we will call it as tetrad. We'll call it as what? Tetrad. How many chromosomes are over here? 4. Right? So I said that I'm starting with what? 4 chromosomes. 2 homologous pairs. I'm starting with 2 homologous pairs. 4 chromosomes. Now, this homologous pair has come close to each other. This homologous pair has come close to each other. Understand? So this is one tetrad, this is another tetrad. Another thing, when 2 homologous chromosomes lie next to each other, this is called synapsis. This is called synapsis. Let me mention over here. Homologous pairs of chromosomes come close to each other and form tetrad. This phenomenon is called as synapsis. Now, please pay attention. Here the spellings of synapsis are different. The word synapsis you learned before in neurons. There it was synapses. It is SIS, but the pronunciation is exactly the same. You see, but plural is synapsis. Understood? So this is synapse. This, you see, when they are arranged together in a tetrad, that phenomenon is called what? Synapsis. Understood? Now, this tetrad can also be called as bivalent. Let me mention over here on a side here that a tetrad, also called as bivalent. When 2 homologous chromosomes, two homologous chromosomes which are in tetrad form come close to each other, that is also called what? Bivalent. Means that tetrad is also called as a bivalent. Understood? Look here. This is one chromosome. This is second chromosome of the same pair, right? Now, there are 4 chromatids, so we will call them what? A tetrad. There are 4 chromatids, we will call them as tetrad. And these are 2 chromosomes near to each other, we will call it as what? Bivalent as well. We'll call it as bivalent as well, yes.
So how does the centrioles make its own copy?
Centriole is one. They just separate from the centriole. Centriole is not some other physical thing. It is just a compact DNA. It's not something else, something other physical thing, right? They are just, these chromosomes, they are just bind at the place of centromere. They are just joined at the place of centromere. Yes.
Tetrad and bivalent are the same.
Yes. But the concept is different. Tetrad means 4 chromatids. Bivalent means 2 homologous chromosomes. Understand? The concept is different. That tetrad means 4 chromatids. Bivalent means 2 homologous chromosomes lying next to each other. Got it? In O levels, you guys learned one thing. That was crossing over. Now, let me ask you, how many brothers, sister you are? 4. Are all 4 of you exactly the same? No. Right? Even though same father, same mother, but you are not exactly the same. Question is why? Question is why? The answer lies over here in this stage of the meiosis, where crossing over takes place. Every time when the crossing over takes place, different portion of the same chromosomes, they overlap with each other. And exchange of chromatids can take place. So every time when a sperm is formed, every time when an egg is formed, it will not be exactly the same as the earlier one, because of the crossing over. Now let me show you crossing over on a side. Now let's suppose that this is one chromosome. This is the second chromosome of the same homologous pair. Same homologous pair. A bivalent, I'm telling again, right? Remember, when I'm saying bivalent, it means homologous pair. Now you understand this, right? And they are close to each other. During prophase 1, what happens? They overlap their chromatids with each other. Like this. They overlap their chromatids with each other. There is a complete chemistry behind it. We are not going into that detail, right? Very complicated. We are not going into that detail. Then what happens? You guys know that in the metaphase, the chromosomes, they align at the equator. We are going towards that stage, right? Then, in the anaphase, they are being pulled apart, right? When the chromosomes are being pulled apart, at that time, what happens? These chromosomes, they exchange their parts with each other. Now, this chromosome will become like this. And this one becomes like this. What has happened? Exchange of the parts of the chromatids. Exchange of the parts of the chromatids. Now, when chromosomes overlap their chromatids with each other, right? When chromosomes, they overlap their chromatids with each other, that stage is called what? Crossing over. Now, let's suppose that over here, it was capital A. Over here, it was what? Small a. Now, what has happened? This one got the small a, this one got what? Capital A. Shuffling of the alleles has taken place. Shuffling of alleles has taken place. When the shuffling of the alleles takes place, every time a new combination will be formed. Every time a new combination will be formed. Now, maybe this time the overlapping took place from here. Next time, maybe the overlapping took place from where? From here. Only from the last part. Understand? So every time there is a new combination. Yes.
So why doesn't this overlapping happen in mitosis?
Yes. In mitosis, it is exactly the same copy. And remember that the mitosis is taking place in the stem cells, other kind of cells. These are special gametes, specialized cells where gametogenesis takes place. So that's why it does not take place over there, right? It takes place during only the gametogenesis process, during the meiosis.
So this shuffling is of random alleles? A,
Yes, random.
A, B.
Yes. Because you see, I told you, right now maybe it is happening over here it was happening over here. So every time when there is crossing over, and maybe not only the crossing over taking place over here, maybe crossing over is taking place over here as well. Sorry. And over here as well. From more than one place.
At the same time.
At the same time. It can be maybe taking place from more than one place. You see, there is sometimes more complicated than this one as well, but I don't want to confuse you guys. I am keeping it simple, right? Understood? So, when the crossing over will take place, new combinations will be generated. So, in the next stage, what I am going to show you guys, I am going to show you two things. Crossing over, right? So, next stage, crossing over of chromosomes. Now, please pay attention. We have red and blue ones, so we will keep on using that one. Understood everyone? Yes.
So why two of them so big and two so small?
Just to differentiate, these are two separate types of chromosomes. Right? So I showed two different pairs, right? I am showing two different pairs, right? And over here we have centromeres which have started producing what? Spindle fibers.
So this all is early process.
No, we have gone into late now. Early prophase were over here.
Only that much.
Now we are very late. Sorry? No. Because of cytoskeleton. It keeps everything in order, in place. Right? Now, next step, the nuclear envelope is completely disappeared. Right? And spindle fibers, they attach to the centromeres of the chromosomes. Right? Next step, what happens?
The crossing over is in late process.
Yes. Nuclear envelope completely disappear. And spindle fibers attach to centromeres of bivalent. Understand? Now I will draw the diagram over here. No more nuclear envelope I am going to draw. Now here we have and understood everyone? Now the prophase is finished. Prophase is now finished. Next stage is what? Metaphase 1. What happens in the mitosis of metaphase 1? Just that the chromosomes they arrange at equator. Right? So, next is yes, they still did not separate from each other. They will become separate from each other when they will be pulled apart. They will become separate from each other when they will be pulled apart. So, metaphase 1, right? Now, in the metaphase 1, we have learned that bivalent arrange at equator, right? Can I rub this? Right? We have understood this. Now, simply we have now can you see I am showing them exactly at the center, right? Sorry? Yes, they are crossed over each other. I am showing that. Still, I am showing that. Can you see?
Not exchange, right?
Not exchange yet. They are still crossed over. They are still crossed over. Remember one thing. Exchange of the parts of the chromatids takes place when they are being pulled apart. Understand? Next step is what? Anaphase. Anaphase is a stage when the microtubules of the spindle start contracting and start the chromosomes to pull apart, right? And this is the stage when exchange will take place, right? So I will mention over here. Next is anaphase 1. I will show the anaphase 1 like this. Now, if you guys remember, in mitosis, the chromatids used to separate. Now they are not separating. Whole chromosome means that whole, you see, this whole chromosome is going into one direction, right? But what has happened, please pay attention, that this blue one is on the top. So the blue one has got the part of what? Red. Red one is at the bottom and he has got the part of what? Blue. Understand? Same thing that over here, the blue one, this is a small one, got the part of red. And the red one got the part of what? Blue. Understand? So what is happening that, let me show, I'm using the green color. So what has happened? That the chromosomes are being pulled to the opposite poles. Let me write over here that microtubules of spindle fiber contract and pull the chromosomes to opposite poles. Understood, everyone? Now, then comes the last stage of the meiosis 1, that is what? Telophase 1. telophase 1, what will happen? The chromosomes have reached to the opposite poles. Chromosomes have reached to the opposite poles. Now, if you guys see that in the anaphase 1, when they are being pulled apart, I have shown that exchange of the chromatids. Right? So crossing over is now complete. Crossing over is now complete. In telophase, the chromosomes, they will reach to the opposite poles, and then what happens? Reappearance of the nuclear membrane. Reappearance of the nuclear membrane, which will be followed by what? Cytokinesis. Which will be followed by what? Cytokinesis. Right? So next diagram which I am going to show you, simply I am going to show the two nuclei. And you are going to understand one very important thing over here. Now look here. Interesting thing. This is the blue. Blue with the red. This is the red with the blue, the larger ones, and this is the smaller blue with the small red and smaller red with the smaller blue. Now look here. When we started, how many chromosomes were there? 4. What has happened now? Chromosome number has become what? Half. Chromosome number has become what? In both nuclei. Next step, what will happen? Cytokinesis 1. Right? You know how the cytokinesis takes place? That over here in the middle there are microtubules which are bind to the both ends, right? They start contracting in the middle, start bringing the cell membrane closer. Again, reference to the cytokinesis of mitosis. Exactly the same procedure which I have taught you last year. Right? So when the cytokinesis will take place, I can show you this in 2 stages. That first, there will be cleavage furrow. I am showing it now quickly like this. And eventually, 2 separate cells are being formed, right? 2 separate cells are being formed. Now, one of these cells have this, and one of these cells is this. Got it now? Now what will happen? Up till here, this is what? Meiosis 1 is complete. We have reached up till this stage. Next, each cell will undergo next division, which will be exactly like mitosis. So I don't need to explain that, right? So what will happen during that? What will happen during that? We have just 5 minutes left or more. 5 minutes, no problem. Tomorrow, if you will ask me, I will draw that mitosis part as well. But you can go and check the video, right? But let me tell you right now, what will happen simply? Just forget that this cell now. Just pay attention towards the one cell. Again, what will happen in the prophase of 2? That will be called prophase 2, metaphase 2, anaphase 2, telophase 2. Understand? In that, what will happen? First of all, disappearance of the nuclear membrane. Second, copying of the centrosomes, traveling the centrosomes to the opposite poles, formation of the spindle fibers. Spindle fibers attaching to what? Centromeres. Understand? Understood? Then will be metaphase 1. What will happen there? Chromosome arranging at the equator. Then will be what? Anaphase. Anaphase 2, sorry, right? During that, the chromatids becoming separate. Here chromosomes became separate, right? Here chromatids will become separate. When the chromatids, they will become separate. So at the end, what will happen? Just let me show you over here. Just let me show you over here. Which kind of 4 cells we will get? Inshallah, tomorrow I will draw that for you as well. Okay? Don't worry. So we will have 1, 2, 3, 4 cells. Now,
But won't it just be one
sorry?
chromatid per cell? So
Yes, 1 chromatid per cell, per chromosome. So one of the cell will have this. Let me show this and this. Other cell will have this with this and this with this. Can you see? Right? So this is the one, right?
yeah, the crossing.
Then other one will have this with this, and the other one will have
There's two different.
this and this. So you can see that, right? 4 different combinations, 4 different types of cells are being formed. 4 different types of cells are formed. Because of what? Because of crossing over. Because of what? Because of crossing over. See, I took, look here. This is the one's chromatid is unchanged. Can you see? Unchanged. One chromatid is unchanged. One chromatid, the chain chromatid. The chain chromatid are shown over here, right? So 4 different types of cells will be formed. Understood? So every time when the fertilization will take place, there will be a different type of egg because of the crossing over. There will be a different type of sperm because of the crossing over. So variation is produced. Understand? So this is the reason that there is variation in the human beings. We are all humans, but we are not exactly the same. Someone is tall, someone is short, right? Even there are 2 brothers, they don't have exactly the same height. Until or unless they are identical twins. Even the identical twins can have some differences among them. Those are because of environmental factors. Maybe one is eating more, other is eating less. Right? So those environmental factors also affect on the variation. Understood, everyone?